before we get started, I'd like to ask everyone to please find the chat window. If you don't see it, you can move your mouse down to the bottom of the screen and there should be a little pop-up toolbar that has a chat icon. And I'd like to make sure that everyone can hear me. So please type a thumbs up or a yes in the chat box so that I can make sure that our audio is clear and getting across. Yeah, great. Excellent responses, thank you. This is our webinar on new and emerging capabilities, and our understanding was from the last webinar that we had when we were discussing the EMSL proposal process that there was a lot of community interest about some of our capabilities and technologies. So I'm going to be introducing some of these and understand that it isn't all encompassing. So since everybody can hear me and you know what the webinar is for, I'd like to say that we're going to have 2 polls. I'd like everyone to participate in this. And the first one is, we'd like to know something about you. So this poll is asking, what position do you currently hold? Please let us know if you're a graduate student, postdoctoral fellow, academic faculty staff, or scientist and engineer in others such as the national laboratories, the industries, or NGOs. While all of you are answering that, I'll introduce myself. My name is Nancy Washton. I am the Deputy for User Programs and the Capability Lead for NMR and EPR. In my role as the Deputy for User Programs, I help coordinate between the EMSL leadership team and the other capability leads at EMSL. And the majority of that work involves making sure that we get your research projects to a point where we provide beneficial data and we end up with high impact publications. So let's see, I'm looking for the results from the first poll. So we have 46% scientists in Industry National Lab Research Institute, 31% faculty and staff, and the remainder is a mix between postdoctoral fellow and graduate students. And so the second poll is what's going to be near and dear to my heart, and that is how many of you actually expect to submit a proposal. And so I hope by the end of this 30 minutes that I will have convinced any of you that weren't committed to doing this, that you will indeed submit a proposal. So while you're answering that, I'd like to point out that EMSL currently has 2 proposal calls open. The first one is for large-scale research, and the due date is March 4th. That is for a 2-year stint with access to EMSL. The second one is FICUS. That is a joint proposal call between EMSL and the Joint Genome Institute, and that due date is March 20th for a letter of intent. Now, I would like to point out that on all of the slides you're about to see, there are names, emails with contact information, but my name is Nancy Washton. You'll see my email. All of the emails will be put in the chat windows so that you can cut and paste them. I want everyone to feel free to contact me directly with any questions, any assistance, anything that you need. I am happy to help you directly or to make sure that I contact you to the right person at EMSL that can address your questions. So let's see what the results were from this. Okay, so we have 38% yes, I applaud that. 54% haven't decided, and so you're my real target audience here. I want to make sure that I reel you in. So thank you very much for answering that. The last thing I want to note is that I will be answering a few questions that you type into the chat window as we go slide by slide. There are 7 total slides. My intent is if I can't answer all the questions because of timing constraints, I will stay online after the official webinar has completed to try and answer your questions. So we're going to get rolling as we move on to our new technologies. So the first new capability is stable isotope probing. Now, you'll notice as we go through the slides that they're all organized in the same way, where I try to let you know what this capability or technique is, the type of information that it provides, and what general areas of research benefit from it. So I think many of you are probably familiar with mass spectrometry techniques. So we are enhancing our stable isotope probing capability, and you'll see a cartoon on the right. And what this shows is using labeled compounds. In this case, it's an example of carbon-13 labeled CO2 as a feedstock for a plant. And we can follow that carbon-13 through the biogeochemical cycle in the soil. In the end, for those of you that might be interested at understanding what microorganisms in the soil are uptaking something that came from the plant and would in fact have that carbon-13 label in it because it passed through the plant, you can determine where it ended up in the microorganisms, such as in the DNA, in proteins, and in metabolites. That'll help you understand the entire biogeochemical cycle. We are very, very excited to be offering this for our current proposal call. So this will be online in the calendar year of 2020. And so this allows time-based or temporal tracking of nutrient flow and fate. And this is of widespread use, not just in microbial ecology and biogeochemistry, but also in what I would consider hardcore microbiology, synthetic biology, and biochemistry. And for any of you that might be looking at contaminants in the environment, it's also useful for that. So are there any questions on this particular slide at the moment? Let's see. I do not see anything, so we'll be moving on to the next new capability. And this one, of course, is near and dear to my heart. This is an 800 megahertz nuclear magnetic resonance spectrometer. And for those of you that are a little less familiar with NMR, it is isotopically selective. So when we run an experiment, we have to choose the isotope of interest. So we could choose carbon or nitrogen or protons. Obviously, those three are very prevalent in biological systems, but also phosphorus. This is a non-destructive and can be rigorously quantitative technique. And so we can look at both solution state samples, such as for metabolomics or solid phase. Now, this new capability is going to be specifically for high throughput metabolomics. So one of the things that's been limiting in our NMR capability here in the past, especially as compared to mass spectrometry, is our inability to have true high throughput. So in the mass spectrometry capability, they have high throughput, they have robots, they have auto samplers where they can go through hundreds of samples, and we have not had that. This new system will allow us a 400 sample well changer, so we can do true high throughput. And in addition, it provides enhanced resolution and sensitivity because of the high field of the spectrometer. So this is going to be particularly important for complex mixtures, not only of metabolite mixtures from microorganisms, microbial communities, but also for things like biofuel production, where you're dealing with these very complex, structurally complex compounds such as lignin. And we're trying to understand how the lignin might change as a function of processing conditions or enzymatic treatment, things like that. And lastly, I would like to point out that in addition to structural information and identifying unknown components, that this technique can also be used to measure dynamics, such as exchange, diffusion, and kinetics. I think most people are much less familiar with that. And so are there any questions on this? I don't see anything, so we'll be moving on to the next one. The next new capability is also something that falls under my purview. And this is an 800 megahertz dynamic nuclear polarization spectrometer, also DNP. We have a lot of acronyms in science. This is a form of magnetic resonance. And when you look at the instrument itself, you'll see a little cartoon on the bottom right. It fundamentally looks just like an NMR. And so what's key here is looking at the plot on the right-hand side. The red line is where we've done traditional nuclear magnetic resonance of a solid phase soil sample. And then we've conducted the same experiment using DNP. And you can see that there is a significant enhancement of signals. And this was a carbon-13 direct detect experiment. And so what we're doing is we're trying to identify in this sample what classes of organic compounds were present. Typically, we're measuring that as a function of environmental perturbation, such as where it is in the core depth. If we have a sample that's closer to the watershed and it's more hydrated than other samples. And so we can start to understand how the organic compounds are breaking down or not breaking down, whether we think they're bioavailable or they're actually passivated and not available to the microorganisms in the communities. And so the difference between traditional NMR and DMP is that this is a surface sensitive technique. And so we do get structural details and identification, but this is going to be only at surfaces such as interface or the traditional surface or on membranes. These experiments are done at very low temperatures, and so your samples need to be able to withstand, and I believe we had a number on here, it is something like 100 degrees Kelvin, right? So these will not be solution phase samples, although they can start out that way, they end up being solid phase. And so this is under my purview, and this is also going to be available. You can request it when you submit a proposal. Are there any questions on this technique? What's the benefit of DNP beyond NMR? So I have to say, it's not entirely clear to me exactly what you're asking, because DNP is a form of nuclear magnetic resonance. So it provides the same type of spectroscopic data. What it does is it uses small organic radicals that actually polarize the surface, and there's a coherence transfer, and that's how it increases the sensitivity of the signal detection. So I would say that since DNP is NMR, that it really doesn't go beyond that. Were there any other questions? Sure, and if you'd like to discuss that in more detail, please email me or just give me a call, and I'd be happy to discuss it, because I'd like to understand more sort of what you're asking. So the general areas of science would be plant structure, so plant cell walls, biochemistry, soil science and biogeochemistry, and also material science and health science. So now we'll be moving on to what we consider to be our emerging capabilities. Now, these are capabilities that are more mature, that EMSL has had for a while, and that were traditionally brought to bear in sort of the materials realm. And perhaps people that are working in biogeochemistry and microbial ecology wouldn't have thought about bringing these techniques to bear on their research questions. And I hope to show you that it's very beneficial if you do this. So the first one is X-ray photoelectron spectroscopy, also known as XPS. And this is a traditional high vacuum surface sensitive technique. We use what's considered soft X-rays. So X-rays, soft and hard, are typically viewed that way in terms of the energy that they carry. And for any of you that have worked at the beamlines, such as the Advanced Light Source or the Advanced Photon Source or Brookhaven, you'll know that depending on what beamline you're on, you could end up with soft X-rays or hard X-rays, and that has a significant impact on the stability of your sample. So if you look at the right-hand side at the plots, this is Elliott soil humic acid. This was gotten from the IHSS, for those of you familiar with soil samples and standards. And so what we were looking at is the utility of XPS to measure what's going on with the soil when it's been treated with NO2+. And this goes directly to the nitrate or nitrogen cycling in soils. Nitrogen is a critical component of plants and microorganisms. They need it to survive. And so we need to gain a fundamental understanding of the nitrogen cycling and inorganic nitrogen cycling in soils. So you can see from the top plot that we had just our baseline unreacted Elliott humic acid. And you can see from the bottom plot that we can clearly measure a new species in NO3. And this is, again, it's surface sensitive, so we usually use solid phase samples, typically soils. We cannot use solution phase samples for XPS, and we can adjust how many monolayers that we're measuring. And that can typically be 2 to 20. The adjustment is done in terms of the angle of incidence of the X-ray on the sample. And so Mark Engelhard is our expert in this area. For those of you that might be interested in seeing some literature, some manuscripts that detail this technique for soil science or other biogeochemical sciences, Mark Engelhard has an extensive list of those and is much more knowledgeable about it than I am, and I encourage you to contact him. Do we have any questions on the XPS? So before we move on, I'd just like to point out that this is quantitative elemental analysis. So for those of you that want to measure the amount of uptake or the amount of loss of some inorganic nutrient or nitrogen or something in the soils, that this absolutely can do that. And the other thing I'd like to point out is that for these techniques, we do not expect our collaborators and our project leads and their team to be able to know in depth any of these. The folks at EMSL are experts. In many cases, they're world-class experts. And we welcome you working with us and allowing us to provide these kinds of data and information for your projects, because we certainly benefit as much as you do from this. So please understand that we are here to work with you as full collaborators on this. So the next emerging capability, oh wait, I think we have a question. Let's see, is there an environmental reaction chamber attached to this particular XPS instrument? The answer is yes. So thank you for bringing that up. We do have effectively a whole daisy chain of glove boxes. So if your samples are air sensitive, water sensitive, just sensitive to anything, oxygen, whatever it might be, we can do aerobic, we can do anaerobic. You could send us your sample in these little sealed metal valving systems. I apologize, I don't know what they're called, but I've worked with them. And we can open them up under a completely controlled environment, and then the sample is put into the XPS and it never comes into contact with ambient. So yeah, thank you for that question. That was very, very helpful. So moving on to the next emerging capability. This one is also traditionally brought to bear on more materials. It is atom probe tomography. Now, I'd like to point out that this is still very much in development, although as you can see from the slide, and I'll explain, we've had some phenomenal results, basically trying to develop methods so that we can use atom probe tomography for what we call soft materials, biological samples, proteins, and I like to tell people that soft materials like polymers, like your skin is a soft material or a polymer. So this is actually a technique that gives you atom by atom 3D imaging. And so if you look at the bottom right-hand side, you will see a colored plot. Those colors correspond to the species listed just above, and it gives you the absolute three-dimensional spatial distribution of these elements in a sample. And so if you look just to the left, there is a black and white micrograph of catalase protein crystals in water ice. And so what you're seeing in that color-coded reconstruction is how this protein, where those species are in the protein relative to the water. So one of the questions that we often have is with proteins, what is the actual structure? How does that protein arrange itself not only in vivo circumstances, but also in situations where it's going to be in water? A lot of our experiments trying to understand protein structure are done, unfortunately, in non-in vivo circumstances. So for those of you that have done protein structure at the beamlines, to understand this, the real question for the community is that structure indicative of what it's like in vivo? And I'm not entirely sure that we know. I think in most cases we don't. So atom probe tomography gives you yet another view of the protein structure. And so we can use these different data sets from the beamline, from APT, and from NMR, because we can get solution phase protein structure, and we can start piecing together what we think these structures are going to be. So this is going to be any environmentally sensitive biological, and geological materials. Daniel Perea is the point person of contact for this particular technique. And this is, since it's under development, I would like to encourage everyone that feels like this may be a useful thing. Say, if you want to understand where the isotope distribution is in a soil or in something like a plant root, work with Danny and he can help come up with a procedure with your input that might be able to answer questions that you simply couldn't answer using any other technologies. And so that's one of the things that EMSL is really phenomenal at, and this is going to be true for any of the capabilities or technologies that we have here. If you don't see that we're doing it, that doesn't mean that it's not possible. So I'd like you to keep that in mind. Do we have any questions? Does the sample need to be conductive? The answer is no, it does not. My guess would be that the protein is probably not conductive. It would typically be considered an insulator. And so Danny's worked on developing these methods so that we can generate these types of data. Can you do UO2 in APT? So the answer is theoretically, you can do that. I'm not entirely sure whether we can do that in EMSL. And the question is if it's below a certain level. And that's something that I'd have to check on. I'm not sure. Considering that this is a non-dispersible sample, I would say it might be possible. So for those of you that aren't familiar, the question really is, can EMSL deal with radiological type samples? And the answer typically is, if it's not dispersible, so it's not a powder, right, not a solution, and it is completely contained in perhaps an epoxy or it's frozen, and whatever we're doing to the sample does not cause a dispersion or any possibility of it getting out, then I would say the answer is probably yes. So just as a quick afterthought, I would say that since this is a destructive technique and the sample itself is actually sort of ablated, that it may be an issue. So we would have to check with Daniel Perea on that one. The next question is, how do you attempt to get atom by atom 3D imaging? The results you showed are referring to molecules' functional groups. Is it a laser ablation type of system? So I would have to refer you to Danny to answer that question. I would say my guess is if it's not actually a laser ablation system, that it's something that functions in a very similar manner. And can you do chromium isotopes? I would say absolutely we can do chromium, and I'm afraid I do not know what the detection limit is. And again, I would point you to Danny Perea to answer those questions. I will tell you that the detection limit is pretty low. Of course, I'm coming from an NMR background where you basically have to have a kilo of material to see it sometimes, okay? So Danny can answer those very specific questions for you. Let's see, was there, okay. So I apologize that I didn't have the answers to some of your questions, but I hope that you'll reach out to Danny Perea on this. This is really an exciting technique. So now we're going to be moving on to data integration and high performance computing. And in these particular areas, I get really excited because the value of high performance computing and these data integrations is something that traditionally has been brought to bear in other research areas, and now we're using it for sort of biogeochemical and molecular ecology, and it's really exciting to show what we can do. So on this slide, what you'll see is in the large rectangular and very colorful box is a molecular dynamics snapshot. And what that is, it is these small organic molecules, and you can see that there is an example of the calcium dipalmitate, and it is on a montmorillonite surface in the presence of soil organic matter. So one of the big questions that we have is, for nutrient cycling in the environment, what ends up being sequestered? So what is adsorbed or taken up or sequestered or passivated by, say, a clay surface or a feldspar surface, something along those lines? Is it no longer bioavailable? And so most organisms need these cations, they need calcium and magnesium. We'd like to know what's happening to them in the environment. So we use both what we would consider classical methods, which molecular dynamics falls under, and also ab initio methods, such as density functional theory. You can see time-dependent DFT. We have experts that are computational chemists that are now working in the area of applying these computational capabilities to more BER and sort of environmental research projects. And molecular dynamics and NW Chem are very complementary. So the molecular dynamics can actually show you some of the structure and the organization, which I know people that are looking at soil organic matter are very, very interested in. The second thing with the NW Chem is that we can actually calculate spectroscopic spectra. And so what you'll see on the bottom right-hand side is NW Chem generated expected calcium K edge from Zanes, which is X-ray absorption near edge structure. And those data, and the empirical data, would come from the beamlines, such as the advanced light source, Brookhaven, the advanced photon source. And this combination can provide spectroscopic predictions for multiple spectroscopies. So infrared, certainly XPS, certainly NMR. So I'd like you to think about all of the empirical data that you may have generated that's really difficult to interpret because it's not a smoking gun. So these computational techniques can provide additional information that allows us to interpret our empirical data more robustly and accurately. And so Amity Andersen is the point person of contact on this. Please reach out to her. She's been working in the more biogeochemical area for the last several years and has some really good insights into this area. So the last slide is going to be on data integration. And out of everything here, I think this is the one that excites me the most. And the reason is because EMSL, we have all of these different spectroscopic and imaging technologies. And I know absolutely how difficult it is to sit down and to have 6 different experts looking at their data, and we're all trying to overlay the data by talking to each other and by looking at each other's computers. This is really difficult, as anyone that's worked in some of these complex areas knows. And so we have taken to visualizing disparate data sets here. And so what you see on the right-hand side is a traditional Van Crevelen plot with FTICR mass spectrometry data. And for those of you familiar with the FTICR and the Van Crevelen, you will see what you typically know with the legend in the upper right-hand side that shows what classes of compounds are in that plot. And overlaid on the plot are data taken from NMR. And so the black open circles with names such as glutamine and phenylalanine, those are data points that we identified the species both from nuclear magnetic resonance and the FTICR data. Now, I want you not to let this by itself constrain you in thinking about this, but think about all the different types of data sets that we generate for you and the people that you know at EMSL and even other places, and realize that we can put together visualizations for any types of spectroscopic data. So Allison Thompson is the person that is the point person on this project. Please feel free to contact her. And this is very much in development. The last thing I'd like to point out is that these visualization techniques are done on the R platform. And so these codes can be provided to you and your group members so that you can play with it and start generating your own visualization scenarios. This is something that we would really like to propagate out in the community because we think this will speed us to results, and we will have higher impact when we can actually put these complex data sets together and pull out more robust interpretations. So, and I apologize, I didn't ask if you had questions on the high performance computing, but I'm looking this. This completes the actual full-scale presentation. Now, this presentation will be available on the EMSL YouTube channel by Thursday. And so I would ask for any of you that found this useful, and I sincerely hope that everyone did find it useful and informative, that you propagate this information out to your collaborators, your students, people in your departments, so that they can see what the possibilities are. And so for things that you didn't see because we didn't have enough time to go into them, feel free to contact me if there's anything on the EMSL website or the capabilities pages that you're curious about. I will absolutely get you in contact with the right points of contact that are experts in these areas, and we can answer all your questions. We're happy to work with you on crafting very viable proposals for the proposal call. And the last thing is, if there are things that you would like to see in the future, webinars on other topics, things that just you're confused about, or it's like a black box and a mystery here, because EMSL can be a bit complicated sometimes. Please let us know this. You can either ping me directly or put it in the chat box, and we will do our best to answer anything that our community has in terms of questions or wanting information. So I think that wraps us up, and I'll say thank you very much for joining us for this. For me, I've enjoyed this greatly. When I think about all of the possibilities that we have here, it makes me want to come to work every day. I just don't lose sight of that, regardless of anything else. And sometimes I just think in my own small way with people like you, that we can move the world forward. And that just keeps me going. So I hope everybody has a great day. And again, feel free to ping me with any questions or comments that you may have. And thank you.